The work I'm going to talk about today is trying to understand from an ecological perspective why sympatric speciation is uncommon in nature, especially in birds. And the birds I'll be talking about today are those pictured here. These are conifer seed-eating specialists known as red crossbills. Sympatric speciation appears to be uncommon in nature, and it's especially rare in birds. Coyne and Orr, in their book, pointed this out by noting that the absence of sympatric speciation in birds is particularly striking because related species often show the sort of niche differentiation that can cause sympatric speciation. And of course, we have many examples of that in birds, including in Darwin's finches, where strong niche differentiation is driven by performance trade-offs associated with the ability to use alternative resources. So what I mean by that is small-billed birds have high performance or high feeding rates on small seeds, but that comes at the cost of low performance on large seeds. And the opposite is true for large-billed birds. So adaptation to one resource trades off with the ability to utilize another resource efficiently. These trade-offs drive adaptive radiation and speciation in Darwin's finches and many systems, but there's an important caveat, which is that these trade-offs are most pronounced in the non-breeding season, whereas the breeding season in many organisms is marked by a high degree of niche convergence on abundant resources that impose weak performance trade-offs. And again, this is where sympatric speciation is most uncommon in nature. If we consider where sympatric speciation is most common in nature, like some phytophagous insect host races, we see that a key difference is that these insects time breeding to coincide with the use of alternative resources that impose strong performance trade-offs, suggesting that variation in the extent of trade-offs during breeding may affect the likelihood of sympatric speciation, which is an interesting pattern, but ideally we'd like a more direct test of that hypothesis, which is what I set out to do with the crossbills. So in North America, we have 10 ecotypes of crossbill, each adapted to feeding on an alternative conifer species. These ecotypes differ in bill morphology, body size, and also vocalizations. And those vocalization differences are handy because that allows you to tell them apart in the field, which is very nice because there's a high degree of sympatry among these ecotypes. Previous work that Craig Bankman has done has showed that if you look at the conifers that these ecotypes specialize on, they impose strong performance trade-offs. And if you go to the Rocky Mountains where I did my field work, you have a lodgepole and a ponderosa pine ecotype that are sympatric, and in most years you'll find them breeding while they're feeding on these alternative resources. But in some years, they converge in their use of abundant resources that impose weak performance trade-offs like Engelmann spruce. So we have dramatic temporal variation in the extent of trade-offs during breeding, which we can use to explore the consequences for speciation. So to do this, I quantified the strength of feeding trade-offs across 10 different breeding seasons just by measuring the feeding rates of these different ecotypes on these different conifers. It's a simple, direct measure of the strength of trade-offs underlying divergence. For each breeding season, I also quantified the strength of 3 prezygotic isolating barriers. So I used surveys of the different crossbills in different forest types to estimate the extent to which there's habitat isolation between them during the breeding season. So that's not a perfect barrier to gene flow because occasionally individuals are found in the wrong habitat type. But because the energetic demands of reproduction are so high in this system, you might expect that those maladapted immigrants might struggle to reproduce in an alternative environment. They might struggle to acquire sufficient resources, which should reduce the probability of gene flow between ecotypes. So I also collected data on the frequency of breeding by the different ecotypes and different forest types to estimate the strength of reduced immigrant fecundity. But that's also not a perfect barrier to gene flow, and occasionally individuals of different ecotypes can breed in the same forest even if the strength of trade-offs are very strong. But these crossbills differ in vocalizations, which plays a role in mate choice, so we might expect there to be assortative mating in this system. So I collected data on breeding male-female pairs to estimate the strength of behavioral isolation in this system as well. And I also summed the contribution of these isolating barriers together to estimate total prezygotic isolation for each breeding season. So what I'm going to show you is the relationship between the strength of trade-offs on the x-axis and the strength of different isolating barriers on the y-axis. I'll note that the maximum level of isolation is 1, which indicates no gene flow between the ecotypes. Across those 10 breeding seasons, as the strength of trade-offs increases, the strength of habitat isolation increases as well. And we see that same general relationship for each isolating barrier and total isolation. So when trade-offs are stronger, reproductive isolating barriers are stronger between these ecotypes. And this makes sense with what we know about the biology of habitat choice, reproduction, and even this somewhat unexpected relationship between trade-offs and behavior isolation can be explained. Briefly, I used field experiments and field observations to show that as the strength of feeding trade-offs varies across breeding seasons, the extent to which these ecotypes flock together varies, which leads to variation in the strength of behavioral isolation. Another important aspect of this system, and I suspect other systems, is that variation in the abundance of resources could affect reproductive isolation independent of the strength of trade-offs. So to picture this, let's just look at the performance trade-off on lodgepole pine, and let's also imagine that there's some threshold of feeding rates that crossbills have to reach before they can reproduce successfully in a lodgepole pine forest. In a low resource abundance year, it might be the case that only those birds with an optimal bill morphology are able to reproduce successfully in a lodgepole pine forest. Whereas if resources were just more abundant and feeding rates were uniformly higher, we might expect that a wider range of individuals could breed successfully in the same forest type, which could increase the probability of gene flow between these ecotypes. And that's exactly what I found. As resource availability increases on the x-axis here, the strength of reduced immigrant fecundity decreases as well, leading to a similar relationship for total isolation. And I'll also note that there are complex interactions between trade-offs and resource availability in driving the strength of habitat and total isolation in this system. The key point is that reproductive isolation is greatest when resource availability is low and trade-offs are strong. I'm excited about these data because I think they help us explain genomic patterns that we're starting to describe in the crossbill system. So I'm showing you genome-wide SNP data, of PCA of those data, for the North American crossbill ecotypes. And what I want you to notice is that there's one lineage, the Kasha crossbill, which is much more genomically divergent from the rest of these lineages. And that's despite the fact that the Kasha crossbill is a very young evolutionary lineage and is sympatric with the two ecotypes I talked about today. What's interesting about the Kasha crossbill, though, is that it specializes on the most stable seed-producing plant in the world, which imposes very strong performance trade-offs year after year. And that stable resource with stable, strong trade-offs and stable resource availability is associated with very stable and high levels of total isolation between the Kasha crossbill and the two ecotypes I talked about today. These data were collected similarly to the ones I showed you today over 6 breeding seasons in this system. Whereas the other ecotypes that I've talked about today experience dramatic temporal variation in the strength of feeding trade-offs and resource availability, which leads to tremendous variation in the strength of reproductive isolation between them, which might inhibit genomic divergence. And I'm particularly excited because I think this supports a basic model that can help us explain why some groups, like some phytophagous insects, are more prone to speciating in sympatry compared to other groups like most birds. I have a lot of people to thank for help with this, including you for listening, and I'll leave you with this summary slide, and I'd be happy to take any questions you might have.